Well, I think
Something's
it's
just popped
gonna
up.
work. Okay,
Something's just popped up in the top right corner saying YouTube.
good. Just need to wait here till. Okay, perfect. So I got that. And now let's do the sharing. Okay, does everyone sees the slide?
Yep.
Looks good.
Awesome. Looks good. Okay, so hello everyone, and thank you for joining us for today's seminar. My name is Adriana Maldonado Chaparro. I'm a professor at Universidad de Rosario in Colombia, and I will be your host today in today's seminar. Well, before we get started, I just want to thank everyone on the panel from last week discussion on the evolution of a research program. It was a really great discussion. I hope that you got a lot of it. And if you couldn't attend, it's on YouTube as well. Second, I want to introduce our speaker for next week, Wolfgang Forsmeyer from the Max Planck Institute for Biological Intelligence in Germany. He will be talking about the honest signal fallacy and the power of advertising. And just quick reminders for those that are new, this is a 45 to 50-minute seminar. And after that, we have about 60 minutes of discussion and questions. Just along the seminar, make sure to have your microphone muted. And at the end of the presentation, you can type in the question mark in the chat, and we will call you for questions. And if you are watching on YouTube, just make sure to write down your questions, and then Lauren is helping me to check out the YouTube channel, and he will relay your question to the host. Okay, so without further ado, David, thank you for accepting our invitation. David got his PhD in biological science at University of Exeter in 2016, and he holds currently a lecturer position at University of Aberdeen in the UK. David focuses, with no surprise, on studying how and why social interactions occur, and how they influence the life of organisms and how they alter evolution of populations. He works on both wild and captive systems, and also in both animals and plants, surprisingly, which is really cool. You might ask him questions about how he works with plants. And he specializes in using analytical methods such as genomics and social network analysis to answer his questions. On top of everything, David has a great publication record with almost 60 articles published in journals from areas of animal behavior, ecology, and evolution. And he also has a great funding record. So congratulations on that, David. David's also very committed to supervision and mentoring. And in general, as he said in his CV, preparing students for life after school, basically. So with no further ado, David, his seminar title is How and When Social Interactions Alter Evolution. So I'm going to stop my share here, and then you can start your sharing.
Excellent. Thank you very much for the introduction, Adriana. Can you all see that okay?
Yes.
Excellent. Yeah, so thanks so much for having me. I really love this seminar series, so it's an absolute pleasure to be asked to present on it as well. So I'm going to talk about some of the work that's been going on at my lab, which I call the Evolutionary Ethology Lab, about social interactions and evolution. So animals engage in social interactions when they fight with each other, when they mate with each other, when they compete in different ways, and when they cooperate. So when I say social interactions, I have a really, really broad definition of what I mean, so that I include all kinds of animals, both group living ones and solitary ones, that can engage in some kind of social interaction. So what I think is particularly interesting about social interactions is that they allow animals to influence each other. So they can do that through, there's a couple of different ways, and these fundamentally alter how evolution can work. So this here is a little diagram showing how genotypes influence phenotypes and then phenotypes influence fitness. And it's this kind of very simple evolutionary map that helps us understand how evolution might work. But when social interactions, of course, the environment is a factor as well, but that less commonly contributes to how evolution works, apart from providing agents of selection. So when social interactions can fundamentally change how this works, because when other animals are a key part of the environment, then the genotypes of a partner individual can influence a focal individual's phenotype through some kind of social interaction. And these are known as indirect genetic effects, this dashed line here, which is in contrast to direct genetic effects, which is how the genotype of our focal animal influences phenotype. Additionally, we also can think about how partner individuals' phenotypes can influence the fitness of our focal individual. And this is known as social selection, again, in contrast to direct selection, which is how the phenotype of our partner is associated with its, a focal individual is associated with its own fitness. So I'm going to talk about these both in this talk. I'm going to start off talking about some of our work on indirect genetic effects. So I don't work on cattle, but I think this is a useful image to use to talk about indirect genetic effects because they're very important in livestock and in group living animals such as these cattle. And also the fact that they're at a feeding trough is significant because we can think about how that resource availability might be impacting how much individuals influence each other and therefore how important IGs, indirect genetic effects or IGs are. So just in case you're not super familiar with these, I'll just go over them quickly. So we think about how any organism's phenotype is influenced by both its own genes and the environment it experiences. And of course, other individuals in that population are also going to be influenced by genes and the environment. Now, if there's some kind of social interaction, then our partner individual can influence our focal. And if that partner individual's phenotype is influencing our focal individual, it means that its genes are also in some way going to be changing the phenotype of our focal animal. So this dashed line here is our indirect genetic effect. Now, these are really interesting because they provide a whole load of extra genetic variation in a population that can contribute to trait evolution. So this means that IGEs can speed up how evolution works if they positively co-vary with direct effects. So if individuals have genes which cause themselves to be large, and those genes are correlated with other genes that cause their partners also to be large, that's a positive covariance, and that can speed up how evolution works. In contrast, we can get IGEs that negatively covary with direct effects. So I have genes to be large, and those genes cause my social interaction partners to be small, then that's a negative covariance, and that slows down the rate of evolutionary change. Now, IGEs have been studied for a long time in livestock because of their close importance for yield traits such as body mass or egg laying. And so in a review in 2014, Ellen kind of summarized the IgE research on livestock and demonstrated they account for between 6 to 98% of the heritable variation in livestock. So that means that IGEs could be an enormous component of evolutionary potential in a population alongside direct effects. So then the question is, okay, that's for livestock. Most of us here don't work on livestock. So what about IGEs in natural populations? Are they significant, as significant in that context? And this is where my work on North American red squirrels comes in, where we were trying to estimate IGEs in this species. So this was part of a really amazing long-term study called the Kluane Red Squirrel Project, which was started by Stan Booten, running from about 1987 until present. Each squirrel can be individually tagged from a very, very young age and then tracked throughout its life. This allows us to measure key life history traits and behavioral traits such as territory ownership, reproduction, and then they're aging from when they're born until they're not seen again, and we assume they died. And that's a pretty reasonable assumption because the study area is surrounded by areas of unsuitable habitat. So if the squirrels are no longer seen in the study area, they most likely have died. So this population is really, really closely tracked, and we can get some really, really high resolution data on when the date a female squirrel gives birth and how many pups she has, and so on. And then we have a pedigree for both males, for mothers and fathers based on DNA analysis. And so we can do some interesting genetics and evolutionary work as well. Now, this species of squirrel emits a very characteristic vocalization known as a rattle, which it uses to delineate its territory boundaries. And so squirrels will go around the trees, rattling where they think the edge of their territory is, and allows them to establish its position in the forest. So we can map the positions of these rattles to work out which individuals are living near each other and which are not. And then we can assume that those individuals living close to each other are interacting, possibly over resources, possibly over space, and vocal communication. They can influence each other as well. And so we can see if there are indirect genetic effects between neighboring squirrels. And the trait we're focusing on is the date a female squirrel gives birth, or a parturition date. We know this is a resource-dependent trait. So squirrels with more resources give birth early, squirrels with fewer resources give birth later. And so it's a good candidate to look at IGEs in. And because this is a study that's been running for over 30 years, we can also do some comparisons. So we can look at years of low density, where we might expect there to be limited social interaction, and so squirrels to have no real strong effect on each other, and IgE should be near zero. Whereas we can also look at high density years where we think, okay, squirrels are going to be influencing each other a lot more because they're literally cheek by jowl, and they should be impacting each other's phenotypes. In particular, I predicted a negative covariance between direct and indirect effects in years of high density, because we expect squirrels who have lots of resources to give birth early, and that will cause their neighbors to have fewer resources and give birth later. So we estimated both direct and indirect genetic variances and their covariance at both high and low density. So at low density, we see a DG estimate of just under 5% of the phenotypic variation is due to that direct effect of an individual's genes on its own phenotype, which is in line with previous estimates in this population. And the IgE estimate was incredibly small. We basically couldn't estimate it. So that means that squirrels were not influencing each other's phenotypes at low density. In contrast, at high density, although we see a very similar estimate for the direct effect of just over 5% of the phenotypic variance, we now see borderline significant IgEs emerging. So squirrels now are starting to impact the phenotypes of their partners. And as we predicted, the covariance is negative. So now squirrels who give birth early call their neighbors to give birth slightly later. Then we can work out, okay, these are nice numbers, but how do they impact the kind of likely evolutionary potential of this population? So we can do that by calculating the total heritable variation. Now, I'm not going to put up too many equations in this talk, but I think it's useful to try to explain this one. So here we have the direct genetic variance, and we're adding to that some the covariance between direct and indirect effects with some transformation to account for the number of individuals. And we're then also adding the indirect genetic variance with, again, another transformation depending on the number of individuals you're interacting. And that's a fairly straightforward sum of a few different parameters. But what's key is that the direct and the indirect genetic variances can only be positive. So as soon as we start seeing these numbers grow, we know that the total heritable variation in the population is increasing, and there's increased evolutionary potential in the population. In contrast, this covariance can, in theory, be negative. Yeah, it may well be positive or maybe near zero, but it can, at least in theory, be negative. And what that means is that this covariance can actually reduce the total heritable variation. So in some cases, social interactions causing indirect genetic effects can completely wipe out the evolutionary potential in a population by having this negative impact. Now, so we can then plug all these numbers in and calculate how the heritable variation in a population changes between low and high densities. So what we see is that at low densities, the heritable variation with DGs alone is just this value here, about 4.6%. But when we're adding in this positive covariance, we're seeing a large increase in the heritable variation. Kind of cosmically, at high densities, the impact is exactly the same. Although we have a negative covariance, which would bring the heritable variation down, this large variation in IgEs essentially then rescues that. And it means in the population, despite this increased competition and this negative effect individuals are having on each other, there's still substantially increased heritable variation thanks to social interactions causing IgEs. So in summary, along with all this work on livestock, we are actually able to estimate IgEs in groups or in territory sets of wild animals. And we possibly saw a more negative covariance between DGs and IgEs at high density in line with our prediction. And the total heritable variation was much higher in this population if IgEs are included, which their social interactions are fundamentally increasing the evolvability of the population, which is super cool. So that work is published in the Journal of Evolutionary Biology. So there I was talking about IgEs from spatial neighbors, so squirrels that are living near each other in space but in the same year. But rather interestingly in the squirrels, we can also think about effects from temporal neighbors. So squirrels in the same place but adjacent years. And this happens because squirrels have an extended phenotype, which is their kind of cone store, which squirrels bury cones in the ground and then eat them over winter. But often a squirrel will die and leave cones in the ground, and the next squirrel can come along and eat those cones. And that fuels their resource-dependent traits. And that means there's a possible indirect effect through this extended phenotype between squirrels that never meet. Unfortunately, I don't really have time to talk about that here, and it's dubious whether that's really a social interaction, but you can read about this effect of this extended phenotype in that paper there. So that's a nice example in squirrels, but what about more broadly? How important are IgEs? So to answer this, me and some friends decided to do a meta-analysis, a lot of it over the COVID pandemic, to kind of see how important IgEs are in more different populations. To a range of questions. So how prevalent are IgEs in general? What percentage of phenotypes do these indirect genetic effects explain in general? And how do those compare to direct genetic effects? Additionally, we're looking across lots of different studies. We can split up the estimates into different categories and see when IgEs are most prominent. So for different trait types and for different classes of individuals, such as age, sex, and so on. Finally, we can answer the question, how do IgEs typically influence evolution? Do they speed up evolution or do they slow it down? And we can do that by comparing the total heritable variation in the population with and without IgEs. And we can also look at the typical sign of the correlation between DGs and IgEs to see if IgEs speed up or slow down evolution. So to do this, we compiled a long list of papers, and we started out with 1,000 papers to go through where we screened the titles and abstracts. Quite quickly, we realized that many of these are not useful for our purposes, and we eventually whittled it down to a list of 47 articles out of that 1,000 that we could use. And this is a reasonably standard hit rate for a meta-analysis. So if you ever start off with a very large number of articles and you feel you're losing a lot when you're doing your own meta-analysis, then don't worry, that's fairly standard. In order to bulk up our sample size, we found papers that had nearly had all the data we needed, but were missing some key bits, and we emailed the authors asking for those data, and we managed to get that for another 23 kind of estimates. And that overall gave us 180 estimates of indirect genetic effects across 19 different species. And so we analysed these data in a series of phylogenetically corrected meta-analyses to answer our question. So first up, what is the overall typical magnitude of IgEs across all these different populations? And the answer is about 3%. So about 3% of variation in phenotypes across all animal populations we looked at is due to social interactions causing the effects of partners to influence the phenotypes of their neighbors. When we compare that to direct genetic effects, kind of as you might expect, DGEs are typically higher than IgEs. So different ways of kind of standardizing or not both DGEs and IgEs, we see that the IgEs and these social interactions are always weaker than the direct genetic effects. And in studies which estimated both of them, we see about a 5 times difference. So the IgEs account for about 5% of the variation, and the DGEs about 25% of the variation. So quite a large difference there, but still IgEs are significantly present in our populations. When we split up our analysis by trait types, we do see a clear difference among trait types in that reproductive traits and traits associated with behaviors have a higher influence of IgEs at more like 7 or 8%, compared to morphological, metabolism, and developmental traits, which are essentially much nearer 0. And we found traits related to survival are intermediate between these, and so we're kind of influenced by IgEs to a medium amount. But when we didn't find any clear difference in IgE strength among studies on males and females across different age classes or population types, so both captive studies and studies in the wild. Some of those graphs look a bit suggestive, but when we dig down into them, it's either very small sample sizes, or in the case of studies on females, an incredibly large number of studies on captive chickens and not much else, meaning we don't think we can make any general conclusions there. Finally, when we look at how IgEs typically impact evolution, what we see is that the overall heritability when we account for IgEs is greatly increased. So a really large increase from typical heritability to the heritability accounting for IgEs and their covariants. And this is in part due to the fact that the typical correlation between direct and indirect effects is positive with a mean of about 0.27, but we do see incredibly large variations. There are lots of traits with a very strong correlation and plenty with a very negative correlation and everything in between. So there's still more to do to work out in what context we might get positive or negative correlations. So overall, IgEs increase the genetic variation available to selection and speed up how evolution works, thanks to this positive correlation, but also because IgEs are felt through the impact on many different group mates, meaning that that initial 3% of variation is felt much more widely. So to summarize, the IgE estimate is small but significant at about 3%, but we see it's more prominent for behavioral and reproductive traits. Interestingly, these are exactly the same traits that often have lower estimates than standard measures of heritability, suggesting that these traits, with a greater emphasis on IgEs compared to DGEs, might be evolving in quite different ways to traits such as morphology. Additionally, we see IgEs increasing evolvability substantially by about 66% because these indirect genetic effects are influencing every individual that one of our animals interacts with, not just one. And we also see a typically positive covariance between direct and indirect effects. So that work was led by Francesca Sanstefano and is now out in Evolution Letters. So I just talked about IgEs, which return back to this kind of map from earlier. The next part is talking about social selection, so how phenotypes of our partner individuals influence the fitness of our focal individuals. Now, I don't work on anything quite as cool as these glorious sea lions here, but what I think is this image helps us kind of reflect on is how it's in many systems where an individual can have a very, very strong impact on the fitness of those it interacts with. If one of these males in this population of sea lions excludes another male from mating, that's an enormous effect of how aggressive they are or their body size on the fitness of their rival. And so their social selection can be really, really substantial in some contexts. So direct selection is when the phenotype of our focal individual is associated with some measure of its fitness. And then social selection is when a partner or partner individuals, their phenotypes are associated with the fitness of our focal individual, mediated through some kind of direct or indirect social interaction. So direct selection is well characterized. We've got lots of meta-analyses and reviews understanding when this is stronger or weaker and different things like that. But a bit more of an open question about the role of social selection in evolution. So I wanted to know, can we use these social selection gradients to capture scenarios where we expect competition for fitness? And then therefore, can we predict when it might be stronger in certain conditions? So at skewed sex ratios or a high density, you might see stronger competition and so stronger social selection. Additionally, we want to calculate when social selection is likely to contribute evolution. And it turns out we need phenotypic assortment for that to be true. So phenotypic assortment is one of the correlation between the traits of our focal individual and the traits of their partner. So where you might get a positive assortment where birds of a feather flock together, so similar individuals hang out with each other, or a negative assortment when dissimilar individuals interact, such as a producer-scrounger dynamic. So we can calculate the overall selection gradient as the direct selection gradient here, added to the social selection gradient multiplied by the measure of phenotypic assortment. And so what's key there is that this product here can end up being positive or negative. And so social selection can increase or decrease total selection. So it can make evolution move more quickly or slow it down, reduce it, or even completely reverse it. So I explored how important social selection was in another animal that lives on trees, not squirrels this time. So this is a New Zealand giraffe weevil. So these are absolutely fabulous insects. So the males can get up to 90 millimeters long. That's a 9 centimeter animal. That is absolutely huge. A lot of that length and where their name comes from is from an extended head, not technically their neck, which they use for kind of jostling on trees and for appealing to females. So Chrissy Painting in New Zealand is in charge of this study system, and she's been studying them for several years now. And in the mating seasons, they can go out, mark individuals on their carapace, and then to recognize them, track their body size, their mating success, their survival, and so on. It's a really, really cool system for exploring some interesting questions. And these weevils gather on tree trunks and compete for access to mates, so they're also a good system to explore social selection. So the data set I used was collected by Chrissy and her student Rebecca previously, and overall there was about 1,200 observations of a couple of hundred males and females mating with each other on tree trunks. So to do a social selection analysis, what you need to do is you need to regress your measure of fitness on both the mass of your individual you're interested in to give you the direct selection gradient, along with the social trait you're interested in. So in this case, the fitness component was the number of unique partners each individual was observed to mate with, which in both males and females is likely to increase their fitness. And we use the social trait we use is the mean mass of our same-sex neighbors, individuals on that tree. I put a little asterisk there because there's a few different studies out there using different ways of trying to measure social selection, but I've done my best to try and pay attention to what the theoretical papers are saying. And as far as I understand it, it's the mean mass of your neighbors or your rivals, not including your focal individual, is a kind of key way of measuring the social trait, and that's what can be plugged into our evolutionary models. So some people are using composite traits or traits that resemble the group phenotype, and ideally you want to be taking a mean of all the other individuals your focal animal is interacting with. So to see when social selection might be stronger or weaker, I also fitted a range of interactions. So I looked at own mass to see if, say, small males might be experiencing social selection differently to large males. I added sex ratio to see if social selection was stronger at male bias sex ratios than when it was even. And I looked at density to see if social selection would be stronger at high densities when individuals might be competing more intensely. And then finally, we measured assortment as the correlation between a focal individual's trait and the social trait, so the mean mass of its same-sex neighbors. So these are the graphs showing the results for social selections. We have females on the left and males on the right. And in both graphs, we've got low density in black and high density in blue. The x-axis shows the mean body size of our rivals, and then the number of mating partners, our measure of fitness, on the y-axis. And what you can see is that black lines are basically flat. So there's no social selection at low densities, and hanging out with large rivals doesn't cause you any problems. In contrast, at high densities, we see negative social selection gradients. So high densities, having larger rivals reduces your fitness and means you're doing less well. And this is true for both males and females. So although the slopes in those two graphs, so the blue lines, look slightly different, the interaction was not significant. So both males and females are negatively influenced by larger rivals at high densities. but we found no interactions between social selection and both sex ratio or own body mass. So these things don't impact the strength of social selection. So now the phenotypic assortment, I just want to remind you that the overall selection gradient is the direct selection gradient plus the product of social selection and phenotypic assortment. And what's kind of key here to note is that because these are multiplied together, if either of these are 0, then the overall term collapses to 0. So if phenotypic assortment is 0, then social selection doesn't actually contribute to evolution. And what we see is that for both males and females at high and low densities, there's no phenotypic assortment. So individuals don't hang out, if they're large, you don't hang out with large or small individuals. It's more or less completely random. And what that means, so to summarize, at low densities, we see direct selection favoring larger body size, which is true in high densities too. At low densities, we see no social selection, whereas high densities, we do get negative social selection in both sexes, but assortment is near 0 in both cases. So that means overall social selection is here and it's representing competition, but it's ultimately not contributing to evolution and it's not changing selection on body size. So that work is summarized in this paper with Rebecca and Christy here. So that's a nice example in giraffe weevils, but what about the overall importance of social selection more broadly? Well, unfortunately, I don't have another meta-analysis for you. So there haven't been that many measures of social selection gradients, and so we haven't been able to summarize them as of yet. In a review paper with Jonathan Pruitt back in 2019, I kind of informally compiled as many social and multi-level selection gradients as I could to kind of see if there were any general trends emerging. So social selection and multi-level selection, which looks at, say, within group and between group selection gradients, are intimately related, and you can kind of transform between the two, which is very useful. So some of the general conclusions I could make was that direct and social selection are often in opposition. And that's seen for traits like plant height, whereas larger plants do better, but they cause their neighbours to do worse through competition or shading. And also think traits like breeding date in both squirrels, for my study, and passerines, where individuals that breed early do better, whereas their rivals therefore do worse. And that's probably because their offspring are competing for access to territories, and so giving birth early means your offspring win, but your rivals lose. Now, like I did with the weevils, some other people had done comparisons across densities, but there wasn't really any consistent patterns emerging. And disappointingly, there was a lack of traits that might be considered cooperative or beneficial, i.e. we're confident that there's a causal positive effect on the fitness of others. So that was back in 2019, kind of we've moved on since then. And so I'm genuinely interested in trying now to look into a meta-analysis on social and multi-level selection gradients, possibly looking at selection on social network traits as well. We're seeing more and more of those being published too. So if anyone wants to join me to carry out a meta-analysis on social selection and selection on social network traits, drop me an email, because I'd love to put a team together and try and make that happen. So what's cool about social and multi-level selection is they allow what I've called opposite responses to selection. So these occur when predicted evolutionary change across generations is opposite, in the opposite direction to the direct selection gradient. So you might see selection for increased body mass leads to the evolution of reduced body mass across generations, or vice versa. And so when we kind of rearrange the equations for predicting evolutionary change, we can arrive at a set of scenarios when this is likely. So this graph here shows the relatedness among individuals on the x-axis, and then the relative strength of multi-level selection, or the kind of ratio between direct and social selection, on the y-axis. And the phenotypic response is yellow if it's positive, which is so in line with direct selection, or blue if it's negative, and so in opposition to direct selection. So what we see is that these opposite responses to selection, this blue area, occurs when individuals are positively related to each other and there's negative relative strength of multi-level selection. So this occurs when you get positive direct selection and negative social selection, which you can think of as competition, so what we saw in the weevils, but also what we might see in squirrels and in the passerines. But interestingly, this is the exact same situation for an altruistic trait. So a trait that has a negative direct selection gradient, so performing the trait reduces your fitness, and a positive social selection gradient. So if other individuals perform this trait, your fitness is increased. So the close relationship between social selection models and models for altruism has been recognized for a while, especially by people such as John McLaughlin. But it's interesting to think about altruism as an opposite response to selection, because you're essentially seeing a trait that's costly evolving across generations. And crucially, both of these require positive relatedness to lead to evolutionary change. So that review on social selection gradients and this stuff on opposite response to selection is published in this paper with myself and Jonathan Pruitt. So along with social selection gradients, IGEs can also cause interesting opposite responses to selection. So this occurs because you might get a situation where an individual with a positive value for a trait causes a reduced trait value in its neighbor. So this would be a negative, direct, indirect covariance. Now, if this individual is favored in the next generation, all individuals might now have this genes to have a higher trait value, this positive effect on their own, but also these negative social interactions are now much more prevalent. And that means that the trait mean may not be changing at all, because although your direct effects have increased, your negative indirect effects through social interactions have also increased, and that can wipe out any expected change. So this is particularly key when traits depend on limited resources and individuals compete for them. So if I have genes that increase my resource share, if resources are limited, that must reduce the resource share for other individuals, as there must be a negative direct indirect covariance. And this is true for traits such as dominance, where only one individual can be dominant, and therefore there's always going to be a negative correlation between whether you're a dominant and if everyone else is dominant or not. But it's also true for any resource-limited trait. So if body mass fundamentally depends on competition for limited resources, there will always be a negative direct, indirect covariance. And this has been well recognized as a major issue in animal and plant breeding, because you can have a very, very expensive, large-scale breeding program that has absolutely no impact on the body mass of your pigs, the height of your plants, because you're not accounting for these negative social interactions caused by resource limitation. Additionally, IGEs can be linked to welfare, because if individuals are impacting the phenotypes of their partners, such as by feather pecking, that can have a really, really strong impact. And so you need to account for how that plays out in your selection programs. Now, we can also think about how these indirect effects can have a strong impact on adaptation. So whether populations have an increased average fitness or a lower average fitness. So if we get indirect genetic effects on fitness, that means that the genes of one animal impact the fitness of their partners. And especially if there's a limited amount of fitness available in the population, such as if territories limit who can reproduce, or there's a limited amount of resource, then we might expect a focal gaining fitness leads to our rivals losing fitness. And therefore, we expect a negative DG-IG covariance for fitness. This actually has really, really big impacts, because normally we might expect that mean fitness increases across generations because it just depends on the direct genetic variation in fitness. This was Ronald Fisher's fundamental insight when he came up with his fundamental theory of natural selection. But what it turns out is that if you have indirect effects on fitness, then you add in this covariance to how mean fitness changes. Now, if resources are limited, this is almost certainly going to be negative. And this can overall outweigh the direct genetic variation in fitness, and can mean that mean fitness can actually decrease across generations during adaptive evolution, which apparently is not really how we think evolution works, but is possible when we account for social interactions into our model of how evolution works. What's cool about this is it separates trait evolution from adaptation, because it can cause mean fitness to not change across generations while allowing traits to keep evolving, such as antler size, weapon size, things like that. And ultimately, this resolves a paradox between Fisher's fundamental theorem and the Price equation, because the Price equation says that if a trait is evolving, there must be genetic variation in fitness. Whereas Fisher's fundamental theorem says if there's genetic variation in fitness, mean fitness must be increasing. Together, those two things imply that trait evolution is always associated with adaptation, with increase in mean fitness, but we know from observations on wild populations that that isn't the case. So IGEs help us reconcile that paradox, and that stuff's published in evolution letters as well. So there's just one more theory paper, I promise. So we kind of extend these insights to show how selection can lead to reduced fitness, and so how adaptive evolution can actually lead to animals becoming less well suited to their environment, which is really not how we think evolution works, but it turns out is possible when social interactions are prominent. So again, we do some rearranging of equations, and what we can see is that maladaptation is more likely when social selection is strong and opposes direct selection. So that's again this blue portion of the graph where we've got negative social selection and negative relatedness. So this most typically occurs when we have positive direct selection and negative social selection, which we can think of as competition. So when animals are really strongly influenced by competition, they can actually evolve to reduce the fitness of their neighbors, and this can lead to reduced population fitness on average. It also requires negative assortment, and that stuff was published in Hereditary with John McLaughlin. So let's get back into the empirical work. So that's a bunch of reasons how social interactions can alter how evolution works and causing, evolution to go backwards, adaptation to not proceed as we should, and so on. So when might these things be more prominent? Well, to work that out, we can look at plasticity in social behavior to see when social interactions are more important and when they're less important. So that's some of the work I've done on dolphins, looking at how they might change their social behavior to get an insight into this. So I don't work on stag beetles, but I kind of wish I did, because aren't they handsome? Unfortunately, this is a detour into the world of marine mammals. So people have been studying dolphins on the northeast coast of Scotland for about 30 years. Aberdeen is around about here, but most of the data collection is by our Lighthouse field station, which is up here in the Moray and Cromarty Firths. So I've been able to use that long-term data set to see how dolphins change their social behavior in response to environmental conditions and food availability. Now, both of these vary substantially over the 30 years we've been looking at this population, giving us a good opportunity to look at changing social behavior due to those two variables. We can also ask, is there individual variation in plasticity? So do some individuals respond a lot and some respond not much? This is important because if that is the case, if you get different slopes among individuals, then in some environments you get very little variation among individuals, and some environments you get much more, and that can be quite important. So I looked at 3 different measures of social network traits. One of these is strength, so how gregarious an animal is. One of these is clustering coefficient, so how closely tied into a clique an animal is. And the third one was closeness, so how well connected an animal is to different corners of the network. All of these were undirected measures, and they were weighted, so they accounted for the frequency of interactions. We defined social interactions in the dolphins, and when we see individuals we can recognize from their unique markings swimming together in a group near the surface. And so we can build up a pattern of who's interacting with who's associating with who over a long period of time. I'll tell you now that we didn't see any response in clustering coefficient to these environmental variables, so I'm not going to talk about it anymore. This is a snapshot of those social networks we've been able to generate over the 32 years of the study to show the kind of incredible variation we see from year to year. There's some results here. We have our measure of the environment, which is the North Atlantic Oscillation Index on the y-axis, and our 2 measures of social networks on the y-axis. And both neither strength nor closeness responded to variation in the environment at either the monthly or the yearly scale. We also saw no plasticity in response to salmon abundance at the yearly level. So we don't have years of different social networks due to years of the salmon abundance in that year. But we do see higher strength, so gregariousness in months of salmon abundance. So in months where there's lots more salmon around, dolphins are much more gregarious. Now, we didn't see any change in closeness in those months, but for both strength and closeness, we do see significant variation in individual slopes. So some individuals are increasing, some are increasing less or not changing at all. So you can visualize those two social networks here. May 1990, a month of low salmon abundance, a much more sparsely connected network, whereas this date in August, much higher salmon abundance, much more densely connected network. So that work was published in Barbaracini in Prop B in 2023. Unfortunately, I discovered an error I'd made in calculating the measures of closeness. And so I had to publish a correction which changed our results a bit, and that came out in 2025. So that's an observational study, and so of course we don't know for certain that it's the change in salmon abundance driving the change in social behaviour. It could be caused by a kind of additional third variable. And so it's great to try and identify causation by carrying out experiments, and often these experiments are conducted in the lab. Additionally, some factors such as humidity are really understudied compared to something like temperature, which is really widely studied. And yet something like humidity is going to be really important for arthropods, because things that are vulnerable to desiccation and water loss might be expected to huddle more closely to avoid water loss when it's dry, and the individuals that don't do that might be expected to die. So we decided to explore how humidity might causally affect social behaviour by experimentally manipulating it and measuring marked cockroaches to see how their social networks changed. So we did this with my postdoc Callum here. So we set up lots of different shelters in an incubator, and in each shelter we fitted a camera that took a photo every minute. Each of those photos was of an aggregation of cockroaches, but then we used a machine vision model, a YOLO V5 model, to extract all the different tags from each of those photos, and so we could see who's hanging out with who. We had over 150,000 images from this study, giving us just over 4,500 measures of individual social networks, giving us a really powerful system to see how conditions experimentally affect social interactions. So we looked at 4 different measures of individual level social behaviour, as well as 5 measures of group level social behaviour, to see if we might see a response at the group level different to the individual level. As you might be able to infer from these graphs, there really wasn't much going on. So the only significant effect we saw was individual closeness, so connectedness to wider corners of a network, is slightly decreased at higher humidities, which is what we predicted, because you expect individuals to huddle less closely together at higher humidities. But obviously, this is a significant effect due to our enormous sample size and biologically absolutely tiny, and therefore we don't think it's that important. In hindsight, we should have varied our humidity by a wider range, say from 30 to 70, but you live and learn. And what was more interesting that did come out of this work was how measures of repeatability or consistency vary between individuals and groups. This graph shows the measure of consistency on the y-axis for different components on the x-axis. So at the individual level, we see intercepts or individual mean values being quite repeatable, whereas groups are not repeatable. So individuals might have consistent social behaviours, but groups don't appear to have social personalities. In contrast, slopes, so how animals change their behaviour across the humidities, showed limited consistency for individuals, but a relatively higher consistency for slopes. So what that means is that some groups consistently increase or decrease their behaviour in response to this change in humidity, but individuals don't do this. So individuals show some consistency in their mean behaviour, but less in their plasticity. Groups, the opposite pattern, limited consistency in intercepts, more so in slopes. So I think this indicates some kind of weird emergent dynamics at the group level, where interactions that are unique to a group lead spiralling off in a particular direction. That is currently under review, but you can see it on bioRxiv. So to summarise the work on social plasticity in both wild dolphins and captive cockroaches, we see that neither species showed much variation in their social behaviour in response to environmental conditions, and so they're either relatively robust or the environmental conditions that were changing were not significant enough. And we did see that dolphins show plasticity to food abundance, as that means that possibly in those months of high salmon abundance, when they're interacting much more closely, we might see significant IGEs and we might get important social selection in those periods. So you might wonder, is this plasticity adaptive? Do dolphins that respond in this way to these salmon abundances, they have higher fitness? And that's a question I've recently explored, again with Barbara, and that's also in review, and you can read that on bioRxiv. Finally, we saw evidence of individual level slopes, both for the dolphins and group level slopes in the cockroaches. And this is interesting for at least two reasons. One, presence of individual level slopes implies there could be genetic variation in plasticity, which means that flexibility could evolve. Additionally, we know for both individuals and groups, there'll be some environments where there's much less variation and some environments where there's much more variation. And that can impact how important selection is and other kind of evolutionary processes. So that's the work we've been up to in the past kind of few years. What is the Evolutionary Ornithology Lab doing now? So one of the things we're interested in, both in the dolphins and the cockroaches, is dynamic network change. So we can look at relatively simple models for individuals preferentially attaching to other individuals and so on, and see if that generates complex network structure. Additionally, we can look at new dimensions of social personalities, such as do some individuals change their connections much more frequently than other individuals? And that's not really captured at all by static measures of social networks. Additionally, I'm also doing some work with collaborators modeling network evolution, and we're interested to see how simple models for selection on network characteristics lead to complex dynamics, including surprising responses to selection. There's interesting dynamics there. And finally, I'm working on kind of feedback between social traits and the social environment. So some measures of social niche construction and how individuals modifying their own social environment can alter how micro and macro evolutionary trends play out. And also to see if animals can modify their environment and this allows them to cope with environmental change. So all of that work has been done with a range of different collaborators and various different funding bodies are listed there. In particular, with squirrels, Andrew McAdam was my postdoc supervisor for that project, so it was really influential. And thankful to Stan Boutin for setting up that long-term study. The meta-analysis was conducted by, led by Francesca and ably supported by myself, Maria, and Alfredo. The Weevils is a kind of project of Chrissy. The theory work was with kind of Andrew as well as John McLaughlin and Jonathan Pruitt. And I promise you, it's much more reliable than any of the empirical work he's been involved with. The dolphin work was mostly with my collaborator Barbara, but Paul Thompson helped set up that long-term study. And the cockroach work with the kind of research associate Callum and some other folks as well. So they've all been instrumental in getting this work out there. So I'm really indebted to them and to the funders. And in my final remarks, I really want to encourage people that we need more estimates of genetic variances and selection gradients on beneficial and altruistic behaviors, because most of these quantitative genetic parameters are in body mass and other things like life history traits and so on. But it'd be really cool if we could get more estimates for things like altruism to better understand how these traits might evolve from generation to generation, how they contribute to adaptation and so on. And finally, I've obviously worked on a range of different species, which has been awesome, but absolutely essential to find good collaborators and enjoy social interactions with your peers and your colleagues, because without that, it's been much less enjoyable and I would have got much less far. So with that, I'd like to thank you very much for listening, and I'm really looking forward to the discussion. Happy to take any questions now or via email. So thank you.
Thank you very much, David, for such a great talk, really. And so we open our floor for discussion. Just a reminder, type in the question mark if you have any question. S if you're a student, so we'll give you priority. FU if you have a follow-up question, so we might just shift the order. I'll try to keep an eye on that. If you're on YouTube, make sure to type in your question on the YouTube chat. We'll make sure to bring it here. And just because, as a reminder, we are building a community, make sure to introduce yourself. Just say your affiliation and what you work on and your name. Thank you. Okay, so we're going to get started with Clara. And David, if you unshare your
Sure.
screen, that will be great. Then we can see each other.
Sure. I'm Clara Jones, Clara B. Jones, Social Evolution, retired. I have a few queries. This is quite sophisticated work, and I want to make sure that I'm clear about certain things. But first of all, I want to ask you whether or not you're using an LLM to augment what you're doing.
Yes.
Do you mind telling me which one you use?
So it's different in the different papers. So some of the IgE work in the squirrels, we had to use ASReml, which unfortunately is a proprietary software used for animal breeders to fit some of the kind of more complex side of things. And in that software, we had to assume normality. So it was a kind of standard linear model. In the work on the weevils, we didn't have a pedigree, so I used GLLM-TMB to fit a generalized linear mixed effect model, using a Poisson distribution for kind of number of mating partners and random effects to control for tree identity, the season it was carried out in, and things like that.
Thank you. Okay, questions. Am I correct in saying that these are epigenetic phenomena affecting gene regulation, and you're not talking about mutations?
So I'm not talking about epigenetics. So what this is additive genetic variants. So kind of we're assuming that there's lots and lots of genes of small effect influencing phenotypes. So it was based on animal pedigrees. And so no, it's not to do with epigenetics. It's also not to do with mutations.
Okay, thanks.
No worries.
Can you give me an example, aside from the obvious, such as infanticide or homicide, killing a conspecific or damaging? Can you give me an example of interactions that might decrease the strengths of selection? I was a little unclear about what you meant there.
In terms of social selection, like how animals influence each other's fitness.
Well, I think you said more than once that interactions could mute or, in my terminology, annul the effect of selection. Is that correct?
Yes. So we can calculate how, say, IgEs, how these variances alter the total amount of heritable variation in a population. And so the way it reduces it is if animals that have genes that have a high trait value, if they cause their neighbors to have a low trait value. So the example is competing for access to resources. If there's only a few resources out there, then if I gain them and get heavy, I'm going to cause my neighbors to be smaller. So that's a negative covariance. And then if selection favors body mass, the next generation is going to be full of individuals with those genes to get more resources and be heavier. But at the exact same time, they're also then, they got the indirect effect reducing the body mass of their neighbors. And so the mean phenotype does not change, because if the amount of resources hasn't changed, then there's no way everyone can get heavier from generation to generation. But the only way that our evolutionary models can make that prediction is if we account for IgEs and this negative covariance, basically saying, along with the increase predicted in the direct effect, there's a decrease predicted by the indirect effect, giving no change overall.
Okay, this is a bit orthogonal, but Are you thinking that differential competition underlies what you're talking about?
Yes, I think a lot of, for there to be variance in IgEs, we need variation of phenotypes, for individuals to be different. And so if to get this negative effect of some individuals do well and some do badly, we must have variation in a key phenotype that leads to competitive ability.
And of course, that's significant when you measure effects in low density versus high density populations.
Exactly, that was the idea. We thought, okay, the
Yeah.
most obvious difference is going to be when everyone's close together versus when they're far apart and not interacting with each
Right.
other. So the first place we're going to look is going to be that high versus low density comparison.
Okay, that's great. And finally, I just have a comment for the students. The first person to introduce social selection to the field was Mary Jane West Eberhard in classic papers in the 1970s, or at least one classic paper. And I encourage all the students to become familiar with that paper. Thanks so much for your work.
Thank you.
Thank you, Clara. Great questions. Michael?
Yes, Michael Dabrowski, University of Bern. Thank you very much. This was extremely informative. I have a sort of general problem or general question that may just show my ignorance. I have the feeling that the two processes that you described, the indirect genetic effects and the social selection effects, are basically just two different levels of analysis of the same kind of phenomenon. The reason why I think this is the case is because interactions, of course, occur between phenotypes. The phenotypes are, of course, strongly influenced by the genotypes, and but also with the indirect genetic effects. I mean, the effects have to be caused by phenotypic interactions. So
Yeah.
I'm not sure whether these two different levels of analysis address the same kind of evolutionary phenomena. What do you think about that?
So I think they are different. I mean, but this is kind of by construction. So if you separate genotypes and phenotypes, then logically you are able, if you want to model it in this way, to separate how genotypes influence phenotypes from your partner and how phenotypes influence fitness. Now, this is just a model, one way of looking at things, right? But what's key is that if you kind of say that genotypes influence phenotypes and phenotypes influence fitness, then you need all those components to then calculate the predicted change across generations. You need to know the genetic variation in traits, which is the variation among genotypes, and you need to know the kind of impact of phenotypes on fitness. And there you need both of those. Of course, you can collapse everything into the price equation, and there are some people who have obviously done a kind of social approach with that, with a within and a between group kind of versions of the price equation. But yeah, so I think this is a way of looking at a complex social world. And I think it's useful because it allows us to make quantitative predictions. But yeah, ultimately you're right. It's phenotypes that interact, and it's not like the genes in one animal leap out and get involved in the kind of developmental process of another animal.
I mean, in effect, I think your answer really points to the to my conclusion that these are two different levels of analysis, and I'm not saying that they are useless or should be condensed to one type of analysis, but it is the same kind of evolutionary process that they are looking at. So it's just two different ways to analyze the problem. It's not that they are addressing different phenomena.
Maybe, but so it's possible that you can get social selection without indirect genetic effects. If like your environment strongly dictates your phenotype, so say your early life effects dictate whether you're large or small or dominant or submissive, that could then influence the phenotypes of partners. And that would be a separate process from genes influencing your phenotype, which then influences the fitness of others.
Mm,
And so I think if you want to capture these distinct scenarios, then I think having them modeling them separately is useful.
yeah.
Just an example.
But still, I think that at both levels, at the level of indirect genetic effects and the level of social selection, you have probably similar influences. As you outlined actually in your conclusion in the end, there may be opposite effects both at both levels of analysis. If I think of your first example, the squirrels, for example, I would also assume that the strongest indirect genetic effects concern mating interactions, for example, probably something with reproduction, like of course at the level of social selection. What would you agree that there are very strong parallels, at least?
Oh, certainly, yeah. Yeah, and of course, and as I said, one of the slides was on IGEs on fitness, right, which kind of summarizes the two processes in one there, where you're just imagining that the genes of one animal can in some way influence the fitness of its partner. So that kind of cuts across both of them in that way. So obviously, yeah, they can be combined. But the kind of the quantitative genetics work always keeps the heritability and the selection kind of separate. And I think it's useful to because both can vary. They might vary, like one can increase and another decrease in one particular environment. So there's some people have done work on demonstrating that direct heritability is large in the same environments that direct selection is weak. And so that means there can be trade-offs there. And so I think it's useful because they can show dynamics that are kind of move in different directions like that. It can be useful to keep them separate. And of course, in one particular study, you may only be able to measure one of them as well. And so it's good to know exactly where that fits in and how that influences evolutionary change. You assume everything else stays constant.
Okay, thank you very much.
Thank you.
Thank you, Michael. Celine?
Hi.
Hello.
Hi, so I'm Céline Teplitsky. I'm a researcher in Montpellier. And thanks a lot for your talks. That was really exciting. And one thing I was wondering about is about when you are calculating the total evolutionary potential and you add the covariance and the variance of IGs. And the thing is that you have to multiply by the number of individuals, and I guess something like a function that says like how much it's like the effect of individual is declining or something.
Yes.
And the thing is that when you look at the dolphins and you say like you have those networks and you really know like how much individuals are interacting with each other, I guess I more or less see, although I'm not completely sure how you actually measure this. And in other wild populations where we don't have, like, you know, you can have like you had in the squirrels, the territories, like how much do you think we can actually reliably estimate those parameters when you want to compare the different types of heritabilities?
Yeah, so we played around a lot with this in the squirrels. So basically the model I used to estimate IGs in squirrels, we borrowed from the forestry literature where they've got trees in a continuous grid in a forest, and they want to know, do neighbors and both the rows and the columns influence the kind of height and the disease resistance of our focal tree. And in most livestock studies, they're in pens. So you just say they're all together, they're all together, they're all together, and it's fine. Whereas in a kind of continuous array like the trees and with the squirrels and with other wild animals, it's not that easy. You can't just say they're a group, they're not a group. The solution are these intensity of interaction factors or these dilution factors, where basically you scale the effect are based on, say, the inverse of group size if your effect is weakened by having more different partners. Or with the squirrels, I did the inverse of distance. You could use something like the association index from a social network and say when this is stronger, we expect more tight interactions. Now, with the squirrels, we played around with a few different options to see what kind of like try out what made sense. So inverse of distance inverse of distance squared, or everyone within a certain distance and no one beyond that, and a few different things to kind of play around with what seems plausible and how that affects the outcome. Alternatively, you could do something based on your data and look at different scales of spatial autocorrelation. So you could see, okay, what kind of spatial or interaction scale do individuals co-vary with each other in some way, and so might be influencing each other, and at which spatial scale do they drop out and stop having an effect of each other? And that may allow you to decide how you're going to scale this strength of interaction. But it's definitely an open question at the moment. We've like barely explored it. And I think, yeah, it'd be good to see more people try it out like we did it with squirrels and see how important playing around with this is and how we might be able to estimate it.
Yeah, because I guess you have like the two issues that you have to figure out, like in which kind of shape it is decreasing, like depending on the strength of your interactions,
Yeah.
but also the scale you're putting on it.
Yeah,
Like-
so we basically with squirrels we tried like inverse of distance and inverse of distance squared, and we decided at a certain distance we were just chopping everything off, making it 0, to avoid it getting like ridiculous.
Okay,
No,
so
we
if-
also tried like a different number of nearest neighbors, so like 3, 4, 5, 6, scaling them all by the inverse of distance and how important that is. So yeah, it's a bit of a, I'm not saying Wild West, but like basically if you've got a good reason to do it, you can go out and do whatever you want, because we just don't know what the right strategy is at the moment. And as long as you're transparent with all the different things you try and we see how that affects the result, I think that's fine. Rather than try everything and then report the best answer. I wouldn't do that.
Okay. Thank you.
Yeah.
Thank you, Celine. So I'm gonna ask my question. Well, I have plenty, but first I wanna just follow up on Celine. And I think just as a comment that defining what is a group exactly, it's becoming more of a methodological challenge. Like even more that we were using group size for many, many more analyses than before, and like trying to understand that what exactly means and how do we measure it and how can we use that measure in a more generalizable way. I think it's becoming harder and something that we might need to probably just slow down and think about it, how to measure this in different species and like with the same sort of sense of what we get. Yeah. And then my question was, so when Clara was asking about the indirect genetic effects, I was thinking, so how do we get the effect? Do we know the mechanisms by which an individual can affect the phenotype of other individual? If it's not epigenetics, then how?
Yes. So the kind of variant partitioning approach, which is what this is, doesn't care about the mechanism. So essentially it's seeing if individuals are associating, and if we know their relatedness, we can calculate how much of the variation in their phenotype is due to their social partners. And then we don't know what the actual interaction is, and that's potentially a weakness of the approach, or like something people don't like, because they can say, oh, you don't know why it's happening, you just know that it is happening. So in the squirrels, we have some theories, like we know their traits are resource dependent. We know that unless there's a glut of resources, which happens every 4 or 5 years, we know they're competing for access to resources, whereas we don't think they're directly fighting each other. So we do think in the squirrels, like it's competition for limited resources. But it's also possible that there's direct communications between individuals that impact their kind of reproduction. So squirrels might be hearing each other communicate, and that changes what they do. So we could go out and test this by kind of doing playback experiments or manipulating resource abundance, something like this, and seeing how the traits we're interested in changing. And that would then allow us to get a better idea of exactly what the mechanism is. You can also do a slightly different approach, which is called estimating the interaction coefficient, and that's regressing a phenotype on a phenotype to kind of see, okay, does the body mass of my rival influence the like growth rate of my offspring, or something, something like that. And you can, or does the behavior of one individual influence the body mass of the focal? And I would imagine you typically only do that when you have a hypothesis. So you think, I think this trait, this interaction type has this kind of outcome. And so you might kind of explore that there. But yeah, so ultimately, yeah, the variance positioning approach doesn't care about the mechanism or is like blind to it. But it's definitely important for making predictions about how it might increase or decrease in different contexts.
Yeah.
Actually, I just thought, just on Michael's point about these processes being similar. One of the reasons why they've been kept separate is they're developed in animal breeding literature, where the selection is entirely artificial. And so that, there's no way in that, or there's limited ways in that case, that these social interactions are impacting who is selected and who breeds in the next generation. And in which case it makes total sense to start with your genetic variance and then just apply selection. LZ in the wild, when it's all about what the animals are doing, then it starts getting conflated, which is maybe more interesting, maybe more difficult. But yeah, possibly the kind of history of the approach in the artificial breeding world is why they were kept separate initially. Sorry, Adriana, I just thought I'd better say that before I forget about it.
That's cool. That's cool. That's awesome. But you did mention something like my next question, it relates to competition. So I think competition, it's coming like every now and then on your talk and also on your answers. And I was thinking if maybe competition for a limited resource will be like a general way to reduce selection in all the systems. Like, can it act in a similar way in every system we look at? And if so, then under our projection of climate change and how things are going to vary in habitats, most likely we're going to get these resources will be limited for a lot of species, basically meaning that there will be an increased amount of competition and then most likely less selection for certain traits.
Well, the selection could still be there, but the evolutionary change wouldn't occur. So yeah, I think you're right. I think I'm like a lumper. I like to group things together and say there are these big general rules. And so I know some people don't. Oh, they love that and they're splitters. But I think you're right. Essentially, if a trait is largely resource dependent, then in order to predict how that trait changes, we don't need to know anything about genetic variance or selection. All we need to know is how is resource availability going to change, right? So if resources decrease over time, then that trait value is going to decrease over time because the trait is entirely resource dependent. The problem is our evolutionary models might say, oh, look, having a higher trait value increases your fitness, and that trait is heritable. So that trait should increase across generations. And then we see it decrease and we're like, what the hell? But the answer is because they're competing for limited resources, and so they're effectively this highest negative social effect increases across generations. And that basically allows us to reconcile our prediction with the kind of very obvious common sense answer that no, it can't evolve because resources are going down or haven't changed. But without social selection or without IGEs, our models might predict an increase over time. So I want to say yes, super general, really important. It's going to be everywhere. I'm sure there are reasons why that might not be true, but yeah, I think competition's kind of general enough. Resources are limited in many systems, and so it must be true that if I gain resources and gain fitness, my rivals lose. So I think it must be fairly general.
Cool. Thank you. Okay, well, is there any other questions? Someone wants to ask any question, or maybe on YouTube? Lauren?
I don't see any on YouTube.
Okay.
Any volunteers to join me on the social selection meta-analysis or on the analysis of social network traits?
I feel that's a great challenge. I would join you, but I already have a lot on my plate right now. But definitely.
Barbara Class has put a thumb up. Yes, excellent.
Yep, I would be up to
you can't actually see because I blur my background, but behind me is a whiteboard with all the different things I'm working on at the moment. I know they're obviously far too long, but one more couldn't hurt, right?
Yeah. So Barbara says yes. should get in touch.
Yeah, for me, that's a maybe, but I would need to know more about what it involves before really saying yes.
Ah, no, come on, no, it'll be fine. It'll be fine. Whatever it is, we'll do. We'll say yes, and then we'll work out what we're going to do anyway. So it'll be cool.
Okay.
So I'm going to take advantage of this. I'm going to ask a last question to give some time to people to make up more questions if they are up to it. So I'm going to shift to the dolphin study where you didn't find any plasticity or social plasticity regarding the Now index.
Yeah.
And that reminds me of social plasticity when I analyze social plasticity regarding food availability as well and other environmental conditions in marmots. And I didn't see any social plasticity either. And I'm wondering if, I mean, given that social interactions are affected by the environment, and we see that in, for example, Lawrence's studies with the primate, with the baboons, why is that we're not seeing this plasticity if social interactions actually respond to changes in the environment? I don't know. Do you have
So
any?
I could maybe start with the dolphins, and then they maybe go more broad. So with the dolphins, this population in the northeast coast of Scotland is actually the biggest population to the body size of this species, and eventually because that's they're the furthest north. So these are like big chunky animals, and we think that, and especially if they like are able to move between like open ocean and the deep sea and the coast, they can be protected quite well from environmental variation. And so I think the dolphins, they're basically robust to this measure of environmental variation. We do know that 2 years after the North Atlantic Oscillation Index changes, the dolphins do change their behavior. But that's because it affects their food availability. So it affects more vulnerable animals. And then those effects knock on consequences up the food chain for changes in the dolphins. But in the current year, in this particular case, I think they're just robust to this particular environmental variable, and it's not going to be severe enough for them to change. More broadly, one potential answer is that the social interactions are too important. Keeping a stable social network is too important, and so it's not going to change. Animals are not going to change it in response to, year-on-year environmental variation. Maybe it's only extremes that are going to like perturb the system enough. So that's one potential explanation. There could be other.
I like that explanation.
Yeah.
That could be something to test in an experimental way. Interesting.
Yeah.
Okay.
Yeah. So you could, for the dolphins, we looked at a kind of correlation between plasticity and fitness, which I didn't present, but that kind of referred to it. And yeah, so you might, if this were true, if social interactions are too important, then you might expect individuals that don't change to have higher fitness than individuals that do change. But that may not be true in all systems. That's just a prediction. So yeah.
Yeah. It links to primate studies where they show that the stability of social relationships are related to increased fitness.
Yeah.
Overall. So I mean, we could probably make a link in a broader scenario, not just dyadic, but just the group as a whole. That'll be, yeah. We've got a question from Clara.
Okay, I have two really, really quick ones, but Adriana, social interactions, if what you mean by that is simply individual interactions between individuals, can be remarkably deleterious, such as manipulation, coercion, exploitation. Those are major categories of interactive effects that are negative. I want to ask Fisher his opinion about something. I'm Clara B. Jones again. Throughout my career, wherever I've talked about abiotic or biotic factors, I have always put a comma behind biotic, and I have always said including social. I include social as an environmental factor. What do you think about that?
Yes, I think on a simple level, other individuals are always a part of, well, they're a part of most organisms' worlds, and so they're a key part of their environment. And so, yeah, you can always think about how those phenotypes are impacting you. The one situation where it gets a bit more complicated than that is if you dictate your social environment. And this is what I want to do in the social needs construction kind of bit I mentioned in my next steps. If I start modifying my environment, modifying my social environment, then it gets much more murky then what's environment and what's my phenotype and my genotype. This then is kind of changing how easily we can predict how evolution works, because suddenly genes influence environment, which influence phenotype, gets a lot more complicated. So I think that's super interesting and kind of getting there. But yeah, it was short answer to your question is yeah, I agree. Like other individuals are definitely a part of our environment.
Okay, that's great. I guess that's it. Thank you.
Grand.
Okay. So if there are no further questions, I think we can just close the YouTube live stream and we can just chat more informally.
If anyone wants to
David
ask
has
like
time.
Jonathan Pruitt as well, I'm more than happy to ask, like kind of talk about that side of things as well.
Yeah. So David, thank you very much for such a great talk and for
worries.
accepting the invitation. And just a reminder, next week, Wolfgang Forsmeyer will be here talking as well about evolution and in social interactions, specifically in the realm